as they say, "Dong." All right. So just to give you a here, this is a video I have just in the module. You know, it's this guy is pretty low tech. It's like some Indian dude who, often just almost looks like he's using a thumbtack to poke his finger, but they're actually pretty good in terms of just showing the basic concepts. So this is just to show you what I was talking about.
Hi friends, today we will try to understand the clotting time, the normally which is around 2 to 8 minutes. The duration when the bleeding starts and up to the formation of fibrin threads is known as the clotting time. In order to do so, clean your finger with sprit and prick the finger. Show that the prick should be bold and the blood comes in the form of drop. Now touch the capillary with the side so that the capillary sucks the blood with the capillary action. The care to be taken so that no air is trapped in it. There shouldn't be any air bubble trapped in the capillary. Now hold the capillary in the palm at the body temperature for around 2 minutes. Then after 2 minutes, break the capillary slightly from one end and you try to see whether there is any thread formation is there or not. If not, then
So there you can see he's actually got a little thread. Like his blood has clotted already here. If it hadn't clotted, you try pulling it apart and it's just like liquid blood on either side. So this kind of gives you a sense of what this looks like. So
you have to break it again after every 30 seconds. You have to need to break it again till you get the threads. Now the time when you get these threads is known as the clotting time. This is how you can calculate the clotting time. Thank you for giving your valuable time.
So just to kind of make it a little more real, like he poked his finger, filled that little glass tube with blood, and started waiting. And after 2 minutes, in his case, it already had a little thread. So for him, he'd have like a 2-minute clotting time. Right, and again, like for the write-up in your manual for this part 4, you discuss what coagulation time is, how we measure it, and also use the values provided in the class data and compare values of subject who took aspirin compared to those who did not. Yeah, again, this is another one of these things. This is usually a really fun lab because everybody gets to like stick themselves and see their blood types and all. So what I'm going to do now is get into the idea of blood typing. And to do this, we actually have to introduce a concept that we're going to look in much more detail once we get into the immune system. And something I'm sure you're all already reading all the time about, just with all this COVID and vaccine and stuff, but the idea of antibodies. In order for blood typing to make sense, and blood type and transfusion reactions and all that, you have to understand what antibodies are. So we're going to first introduce antibodies. Antibodies are also called gamma globulins. They're also called immunoglobulins. And they're basically proteins. And they're often, they're found in your plasma. In our case, when we're going to be looking at them. So antibody means so the antibody is looking at the idea of antigen. So first we need to look at what is an antigen. Antigen means antibody generating, and it is just some shape off of something. When we're talking about coronavirus, the coronavirus has the little spike proteins, right? Those spike proteins could be antigens, some shape that we can recognize. An antibody to the coronavirus is going to, antibodies have, we call a variable region. So this is my antibody here. An antibody has this region that fits like a lock and key and recognizes and binds to a specific antigen. Or say recognizes and binds. So I can have an antibody here that binds to this distinctive spike protein on the coronavirus. This is what you're hoping to get from your vaccine at the end of your month and a half. You hopefully got a bunch of these floating around in your body. You're also initiating other parts of your cell-mediated immunity, which we'll talk about later in the semester as well. But antibodies are just things, these proteins that your body makes. They have this variable region that is going to recognize a specific shaped thing and grab onto it. So are there any questions about what is an antibody? It's a little protein, in this case in our plasma.
Isn't also a protein?
What's that?
Is an antigen also a protein?
The antigen?
Yes.
Yeah, an antigen can be anything though. An antigen is just whatever the antibody is recognizing.
Okay.
So antigen is more generic, and in fact, you can have one antigen that has many kinds of I'm not going to go into so much detail, but let's say, here's some nasty little thing that's going and attacking you in your body. You can actually have different antigenic determinant sites. Like I can have an antibody that actually recognizes the horn. That will recognize this little guy. Or I could have an antibody that recognizes the claw. Or I could have an antibody that recognizes his tail. So you can actually have more than one type of antibody that are going to recognize the same antigen, the same little thing that's generating this whole response. When people talk about polyclonal antibodies, that means you have a variety of antibodies that all recognize the same target, as opposed to monoclonal antibodies, which are all just the exact same copies of themselves. Okay. So now we're set. So the red blood cell. So we talked about membrane proteins, right? Membranes have all sorts of proteins. Blood cells, red blood cells have these membrane proteins, these little marker proteins that can be A-type proteins. And I'm gonna, for the purposes of making my life easy, whoops. I'm gonna say the type A marker looks like that. Oh, it looks like an A. Like when I say somebody is type A blood, I am saying that their red blood cells have this membrane protein that is this specific shape, specific kind of antigen that we're calling type A marker. If somebody is type B blood, they have a different shape marker. They have a shape marker that, for the purposes of simplicity, I'm going to make it look like little bees. They have like the B shape marker on B shape blood, on type B blood. If I'm type AB blood, I've got A markers and B markers on my blood. And if I'm type O blood, I've got nothing. So type O blood is just no markers at all. So this is the first step of understanding the blood typing, is thinking about what are the markers on the blood cells themselves. And then the other thing we need to think about, it turns out your body will actually create antibodies that recognize and attack the marker that you do not have. Right? You don't want to have antibodies that attack your own blood, because then you're gonna be dead. But if I am somebody who is type A blood, I am going to actually have anti-B antibodies in my blood, in my plasma. If I am type B blood, it's going to be the opposite. I'm going to have anti-A antibodies in my plasma. If I am type O blood, what am I going to have?
None.
Now remember, you're going to have antibodies against everything you don't have.
So against A and B?
Uh-huh. What about AB blood?
O.
Remember, O isn't anything, though.
Are you not going to have antibodies?
Exactly. So type AB blood will not have any antibodies. So now let's talk about blood transfusions. When you think about blood transfusions, you think about the blood cells entering into this person's body. Don't think about donating plasma or antibodies. The antibodies are there in the recipient. And you have to think about, what is this recipient's blood and antibodies going to do when they see this donated blood cell come into the scene? And that's how you're going to figure out like who can get what kind of blood. Like if I am type A blood, what kind of blood can I receive as a donation without attacking it?
Another type A or
Exactly.
type O.
Exactly. I could receive A type blood because I'm not attacking A, and I can receive type O blood because there's no markers on that. If you gave me B or AB, both of those have B on it, so my anti-B is going to attack it. I'm going to have a transfusion reaction. Typically what happens is the cells lyse and you spill out hemoglobin and clogs up your kidney tubules, and it's a disaster. Who could I give my blood to?
To another type A?
Yeah, exactly. I could give my blood to another type A. Who else could receive my blood without having a problem?
AB.
AB. AB has no antibodies to fight A. So if you gave my blood to the B person or the O person, it would be ugly because their anti-A antibodies would attack my blood and kind of, break it down. Similarly, B's going to go into where? Who can B donate to?
B.
Yeah, B and
AB.
AB. Who can AB donate to?
AB.
Yeah, just AB, right? If you put AB in any of the other persons, they're going to attack because AB has both A and B. Who can AB receive blood from?
O.
All of them.
Yeah, all of them, right? It has no antibodies, so it's not going to fight anybody. This is what we call the universal recipient. Which is a good thing because AB is very rare. It's only a few % of all the blood types. So if AB was relying on another AB for a transfusion, they'd be in trouble. How about O? Who can O donate to?
Everyone.
Everyone.
Everyone. Since it has no markers, this is our universal donor. And who can O receive blood from?
Only O.
Only O, right? If you get anybody else, they're going to attack it. Again, luckily, O is the most common blood type. So it's likely that you're going to find a donor that can give you O blood if you need it. But O can only receive O blood. So this explains like A, B, and O, and then obviously people usually talk about A positive, A negative. So let's add that into our picture. And that's called RH factor, or sometimes they call it D antigen. So RH factor, it's just another protein marker on the cell membrane. And if they got it, you're positive. If not, you're negative. So if I just have A markers, this would be A negative. If I have A markers and the little RHs as well, this would be A positive. If I'm just a cell without anything at all, that would be O negative. If I'm a cell without any A or B, but I do have the RHs, this would be O positive. So that's all the positive or negative does. It's like, do you or do you not have the RH markers? The reason why we separate it out from the A and the B thing is you don't have automatic anti-RH antibodies if you're negative. Like this person here doesn't necessarily have anti-RH antibodies. So you could actually donate this blood to this person the first time and it would be okay. So you could do this once. But once this person has been exposed to this blood with the RH, they then have a immune reaction and they then create anti once, but then create anti-Rh antibodies. So once this person has anti-Rh, if you try to do this a second time, it's bad, because now they are going to react against this positive blood. So the way Rh is different from the A and the B is the first exposure to that antigen is okay, but subsequent exposures are not okay. The reason why this ends up being really important is in childbirth. Like if here's,
That's why we give a rogam injection in a person's first pregnancy.
yeah, exactly. So rogam, we'll talk about in just a second. Like, so woman is pregnant. Let's say she is A negative. You know, the baby can have any kind of blood type based on, you know, the genes with the dad. Let's say the baby is A positive. It's not a problem if she's doesn't have any anti-Rh antibodies. It's all good, and the mom and the baby's bloodstreams don't mix. But there's going to be childbirth, and it's ripping and messy, and the kid's finally out. But during childbirth, the mom probably was exposed to the fetal blood as this all happened. Which case she would have made anti-Rh antibodies, and if she ever had a second pregnancy with an Rh positive baby, her body would attack it. So this rogam shot, which she gets, this prevents the formation of the anti-Rh antibodies. Right, so if she has a second pregnancy that's also an Rh positive baby, it's not a problem. She's not going to attack the developing embryo. So, but if, you know, before we did this, there was always a danger that if you have a second pregnancy with an Rh positive baby, that your body would attack it if you're Rh negative and you've been exposed to the Rh factor. Does that make sense? So to do blood typing is pretty simple. All you do is we have these little sera that contain the antibodies, and you add the antibodies to your blood and see if it grabs on and coagulates, right? If I am type A blood and I have anti-A antibodies around, those antibodies, whoops, daisy, grab on. So all these blood cells start all getting clumped together. We call this agglutination. And you can see it. So if you have the antibody mix with the blood that has the antigen on it, you can actually see it just visually pretty easy that there is this agglutination, this clumping. So if I take my blood, the way you usually do it is take a slide, label one side A, one side B, have another slide, which is going to be for Rh. And typically you use like a wax pencil to make like a nice circle to keep the blood from going off the edge of the slide. You're going to put blood on it. So this is just a little microscope slide. And then I'm going to add And then I'm going to mix it up and I'm going to see what happens. If I had type O blood, what would I expect? If I had O minus blood, what would happen? What kind of markers does O minus blood have on it?
None.
None at all. So if I add any kind of antibody to it, am I going to see anything happen?
No.
No.
No. If there's no antigen for the antibody to recognize, there's not going to be any agglutination. So if I'm O negative, I should just see the blood stay looking normal. What if I was O positive? Are the anti-A or the anti-B going to have any reaction?
No.
No, those are just going to look normal. What about the anti-RH?
That would.
Yeah, I'm going to see this kind of clumping happening because the RH is going to get recognized by the anti-RH, and we're going to have agglutination, that clumping. How about me? I am type A positive. What's it going to look like if you put my blood through this test?
There would be one for the anti-B, you would see it. And then also the RH.
So if I'm A positive, what kind of antigens does my blood have on it?
A.
It has A. So which of these is going to react with the A on my blood?
A.
The A. So if I'm type A, I'm going to see the agglutination with the anti-A, right? Because this is not my plasma. This is like a drop we're adding from the outside.
What does it look like when it undergoes glutination?
You will see in just a few moments, actually. It looks basically kind of clumpy as opposed to just kind of smooth and red. But you'll actually see some actual pictures of it in just a few moments. So are there any questions about this?
I have a question. I just want to clarify. So if you have A positive blood, you can receive blood from an A negative person without an issue.
Correct.
Okay.
Right,
Okay.
because A negative means that they have no RH, so there's nothing to even worry about.
Okay. Okay. And the same thing with the pregnant woman. If she had like A positive blood, there would be no issue. There would be no
Yeah.
need for the rogam, right?
Yeah, the rogam is
Okay.
only going to be if you are negative, in which case you have the potential to form the anti-RH antibodies.
Okay. And so then your A positive blood would react with the anti-A. It will clump, but
Uh-huh.
not the anti-RH.
No, A
Or
positive.
yes, right? Yeah, yes, with the RH, right? Okay.
Clump with the anti-RH as well.
I think the word anti is messing with me. Okay.
But anti means that it
Yeah.
is against, it grabs it and clumps.
Got it.
And in general, the RH one is a little slower. You have to like heat it up for a minute, and the reaction isn't quite as strong, but you can still see it fine. The last thing before we do this little quiz, again, if we were actually doing this in person right now, I would have been just going off on universal precautions and bloodborne pathogens and stuff. We'll talk more about it, but like right now, in the interest of time, I'm just going to briefly mention the word sharps because that's on the quiz actually. Sharps are referring to anything that's contaminated with blood that can puncture the skin. So, sharps, for our lab today, would have included, the little lancets, right? The things we use to actually poke our fingers. It would include the slides, the microscope slides, because they're made out of glass, the capillary tubes. Because those can puncture you. Wait, not slices, slides. It would include, you'll probably include the toothpicks that we'd use to mix things up, because those are kind of sharp, right? So not cotton, for instance, right? It takes special, your lab, the people who are dealing with the sharps, needles and things contaminated, you have to be very careful with them, and it takes time, and you have special containers. So you don't want to just throw general trash in the sharps containers, because it's a waste of, these expensive resources. So you only want to put actual sharps in the sharp containers. So these go in what are called sharps containers. Which then, whoever's going to be dealing with sterilizing them knows they got to be careful. Like should be slides, not in the trash. You really want to pay attention to you want to keep track of where your sharps are. And you want to make sure that you don't just put them in the trash, because then, it's just some custodian who is emptying the trash has this little thing that's got contaminated blood that can slice through the bag and go through their skin. And all of a sudden, he's got some deadly disease. He's got hepatitis or AIDS or whatever. Right? There's a lot of bloodborne pathogens, things that can make you really sick or can kill you. And you really need to, it's not just like bad technique to take a sharp and put it in the trash. It's like, unethical, because you are putting some random person, at deadly risk of some disease. So you always need to kind of keep track of where your sharps are and make sure they're properly disposed into the appropriate sharps container when you're doing blood work of any kind. As opposed to like squishy things, that you can, we usually in the classroom will have bags that get sterilized, but it's not these special containers for sharps. They're more these bags that get autoclaved, get basically sterilized. But squishy stuff, right? The lab tech or the custodian, can grab this thing and shove it in some place, and there's no worry about some little needle poking through and jabbing them. So, all right. So what I would like people to do right now, I'm going to share screen. So after we're done with this, you can watch this stuff about blood typing and hematocrit. This is showing you what the two different kinds of capillary tubes look like. You should be aware of this. Like on a lab exam, I could ask you, which one would you use, the one on the left or the one on the right if you're doing hematocrit?
The one on the left.
Yeah, the one on the left, the one that's heparinized, is for the hematocrit because it'll keep the blood from clotting. The one on the right is the one you would use for like clotting time. So this blood work typing quiz, you can just go into it. And I want you to do, we have one, let's see, let's just go, I'll put you in little breakout rooms, like do it together. You reach a consensus before you pick your choice for each one to make sure everybody is kind of talking to each other. And we're going to do this and we'll come back here in like 10 minutes. So I would like everybody to do this in their little breakout rooms, and I'm going to set you up right now. Okay. So again, I think it's worth just looking at this little video here, which basically shows you more of the detail of how you actually stick your finger and do everything. Although like on an exam, you'll be more responsible for like interpreting like kind of what you saw like on that quiz. There won't be details about which lancet should you use or something, or which finger should you stick or whatever. So don't worry about that. Definitely you should understand the difference between these two different kinds of tubes. Again, so I said for the write-up, obviously we're not typing our own blood. If you know your blood type, follow the lab manual. Part 1, step 6, describe what you'd expect to see. Draw, do a little drawing of what you would think it would look like if you did your blood with this test, as well as appropriate donors and recipients. Who could you get blood from, give blood to? Again, if you don't know your own blood type, write about me, because why not? For parts 1, step 7, use the class data provided, which I'll show you in just a moment. For hematocrit, like I said, you're not going to actually do it, but discuss hematocrit values provided in the class data, which I'll show you in just a second. Hemoglobin content, I didn't actually talk about this. This is basically a way of trying to figure out how much hemoglobin is in your blood. It's basically another test to see if you have anemia. It's usually related to hematocrit because the more red blood cells you have, the more hemoglobin you got. The little test that we use, this is not how you would do it in a formal lab, in a clinical setting. They actually have these cool things called hemoglobinometers that use light absorbed by hemoglobin and stuff. This is like putting your blood on a little piece of filter paper and letting it dry and comparing the color. But this little thing here, this literally lasts 1 minute and 34 seconds. Kind of watch how they check their hemoglobin content by matching the color. And then there are values in the spreadsheet that I'll show you, and just discuss if those values that you see for male and female are consistent with the averages that I talk about in the lab manual. Coagulation time, we've already talked about this. And then here is the spreadsheet. The spreadsheet basically, open it up more properly. It has here the blood types, male, female, I've sorted already. We have average hemoglobin for male and female here in this grams per deciliter. Here is the hematocrit, male and female. I should make that as percent. So use these averages here to compare to the averages, and the US average that I put in the lab notebook, and you can just comment if this particular class here, spring 2018, seems typical. Also, clotting time without aspirin, clotting time with aspirin, the average. So you can talk about, for this class, how aspirin seem to affect clotting. So again, in your write-up, just make sure you use the data that's available here and kind of compare this spring 2018 class to, kind of more expected averages of things. Submit PDF of Bloodwork lab. And again, this is another one of these things where I wish you guys could be here for this lab because it's a fun one. And like I told last semester as well, if you're on campus when we're finally back in person, like I'm happy to hook you up with some lancets and capillary tubes and little sera if you want to mess around and check your blood type and see how this all works. So we have stuff to spare, so we can make that happen. But otherwise that's it for today. This lab is a lot more, makes it a lot more interesting when you do it in person. But I think it's important information to understand blood typing. Hopefully you walk away from this having a better understanding about transfusions and who you can get blood from, who you can give it to, and why that's the case. So otherwise, I think, the next lab as well there's nothing to prepare.